Hi, good evening. Thank you so much for the invitation. It's a great pleasure to share some of our experiences and the lessons that we learned at the Drug Discovery and Development Symposium. Louise and I are doing this together. We didn't have a practice run, but I think we know each other well enough, and so that we can make this work. I guess I'll start off. Louise, is that okay with
Yeah,
you?
that's a great idea.
Okay, and then you have clearly indicated where you wanted to make some comments on
Yeah,
on the topics that I will discuss first. But whenever you want to say something at other moments, just please fill in wherever you want. And I think also for the rest of the audience, for me it's okay if questions are asked during the presentation, but after the presentation is fine too. But I have no objection in answering questions during the presentation.
I think it's more handy to do it afterwards because it's recorded, so people will do
Okay.
it back, and then it's more, it's a little bit chaotic if it's done in between. So
As always, you are smarter than me in things like this. So
no, I'm not, but I just know it's recorded. So
okay. Can I get the next slide? Yeah. So I will just go through the program and discuss every topic that was during the meeting. So we started with of course a brief introduction by the current president of the PVRI. The PVRI is an institute that is out there for I think more than 15 years now, dedicated to promoting research and education in the field of pulmonary hypertension. Every year there's an annual symposium for all members, and every 2 years there's a so-called drug development symposium. And this year we had the opportunity to host it in Amsterdam. It was nice weather, which is not a given in early June, but I think it was appreciated very much to be able to do this meeting in Amsterdam. So of course, already discussed a little bit. The big change in the care for patients with pulmonary hypertension is sotatercept. It was approved by the FDA and the European Medicines Agency. Unfortunately, it's only available at this point in a couple of countries, but everybody is well aware that treatment algorithms will change with sotatercept being available, and also the ways future drug development will be done will change when patients already have the access to sotatercept. As I said, the drug is only available in a few countries at this point, and part of this has to do with the different processes and the different evaluations that were done by the regulatory agencies in different countries. So we were very pleased to have two regulators present during the meeting. Mitchell Psotka from the FDA in the United States, and Ilana Meurs, who works at the Medicines Evaluations Board in the Netherlands and was involved in the EMA process. And they give us some perspectives on the regulatory process in those two instances. So the FDA made the conclusion that the label for sotatercept was very broad, not just for an improvement in exercise capacity, but also improvements in real clinical outcomes. Whereas the EMA judged it a little bit differently and only gave the label of improvement of exercise capacity. And it made the situation in many European countries a little bit difficult, because this is a restriction of the label and gives other ideas about reimbursement. For example, in the Netherlands, there's still a lot of discussion on reimbursement of sotatercept. And part of this has to do with the different evaluation of the two regulatory agencies. Well, it was very interesting to hear them discuss and to see how they evaluated the same studies in different ways. This is of course always the case when you talk about science. There can be disagreements. I think it is likely that at some point the EMA will also come to a more broader label for the use of sotatercept, but they needed the additional studies that were done in the past couple of months and were finished in the past couple of months to really be able to give such a broad label. Both agencies did a fast track procedure. So initially it went really quite fast, but whether they will be able, like EMA, will be able to make an adjustment in an equally fast way, that is of course the question. So this is still work in progress, and also depends on evaluation of very new studies that were published with sotatercept. Then we had actually I think I should already go to the next slide. Can you give me the next slide, please? Yeah, sorry, I forgot to mention this before. So we can already go to the. I discussed this. We can go to the next slide. I think Louise has her perspective on the matter.
Yes, I do. Well, the main issue is indeed that the final improvement by the EMA was narrowed to improving exercise capacity only, so removing all references to secondary endpoints, and also requiring combination use with other PAH therapies, as in research was also, of course. But what stands out from the patient's perspective is the lack of consistency in how health-related quality of life has been measured across all clinical trials. So not only this one, but in general, because many different prompts have been used, but they are not comparable. And as a result, it becomes very difficult to demonstrate statistically a significant improvement in quality of life, even when patients may actually feel better. And for patients, quality of life is not a secondary concern. It is often our main concern. We don't just want to survive, we want to live. It would therefore be very helpful if the field could agree on more suitable and pH-specific prompts to use consistently in future trials, especially when evaluating new treatments like sotatercept.
Very good point, Louisa. I echo that. Can I get the next slide? Yeah, you went to, yeah, this is the first one. So one of the very important things is, of course, we have guidelines, we have a clear treatment algorithm, but now sotatercept is being accepted, and where to place it in the treatment algorithm? Do we use it in patients only with the highest risk or also lower risks? Do we use it in patients who are just recently diagnosed, or do we wait? A lot of this depends on how the different studies were done, and the first idea of how a new treatment algorithm could look like was published last year. It was a result of the World Symposium on Pulmonary Hypertension, and pretty much based on the results of the STELLAR study. So that was the first phase 3 study on the use of sotatercept in PAH patients, and in that particular study, the patients that were treated with sotatercept were prevalent cases, so patients that were already known with the diagnosis for on average, I think it was about 8 years, and patients that had been using background therapy for a very long time. So when you look at the results of the STELLAR study, the main place for a drug like sotatercept would be in patients that are not achieving a low risk status at follow-up. So the placement would be not at the beginning of treatment, but at follow-up. Patients that are not responding properly, don't achieve low risk. In these patients, sotatercept could be considered on the basis of the STELLAR study. But that is, of course, still a very limited scope. Some patients already arrive at the hospital at initial diagnosis in a very high risk. And should we also consider sotatercept in these patients? Well, now we have the results of the Zenith and Hyperion studies. So Zenith were patients in high risk, and Hyperion were patients with a relatively very recent diagnosis. And this could really change a future treatment algorithm further, and could lead to consideration of sotatercept maybe even before the use of prostacyclin, parenteral prostacyclins. But that is something that a lot of discussion still needs to be done around. The other important point that Luke Howard made was that, of course, we are talking about low risk and as something that should be acceptable, but still patients in a low risk status do have a considerable that is not no risk. Patients still have a chance of to die within 5 years of about 3%. So that is still something that we should not be just complacent about and should just accept, but we may try to achieve even more. But how do we test this in clinical trials? That is very difficult. That means that we need different endpoints, and maybe we have to look back at hemodynamics to really determine whether sotatercept in a low-risk patient still has some benefit. And yet, the other important thing that still needs to be worked out in the future is whether sotatercept will allow for withdrawal of certain treatments. It's already been done, but we still don't know whether this is really safe. But some patients on parenteral prostacyclins responding really well to sotatercept are taken off the parenteral drugs, but there are no really studies that really show us that this approach is safe on the long run. So that is something that really needs to be tested. So a lot of discussion on what the treatment algorithm should look like, and also the realization that actually for the current treatment algorithm, some of the scientific evidence is not even as strong as we might wish, and some of the evidence of sotatercept may even be stronger than some of the evidence that we had in older studies. So I think the next slide.
Yes, actually, I came to the same thing. First of all, it's very encouraging to see that during the World Symposium on Pulmonary Hypertension, sotatercept was discussed as a potential option already in the intermediate low category, after follow-up, of course. For patients diagnosed at high risk, the first step will still be triple therapy with prostacyclins. And during the presentation of Luc Hauer, the possible option of starting with quadruple therapy was mentioned, with an extra option to stop later with the IV and replace it with selexipag if needed. As patient advocates, I truly hope that future evidence will also support the immediate use in patients diagnosed at high risk. So triple therapy, but then including sotatercept instead of prostacyclins. If sotatercept could help delay, or in some cases even avoid the need for IV or subcutaneous prostanoid therapy, that would be a meaningful development for patients. This mode of administration, while effective, places a significant burden on daily life. It impacts mobility, work, social participation, and mental well-being. Any treatment that could postpone or reduce the need for such invasive therapies would represent a real improvement in quality of life.
Yeah, that's an absolutely good point. And unfortunately, the studies that were done so far were always in patients already on background therapy. So it's still a very difficult decision to make on the basis of the evidence. But of course, the benefit of such an approach is very clear. Yeah.
And maybe it's also in discussion with the patients themselves. So you can discuss it as a doctor, you know, that the patients know the risk of doing it, because no future evidence is yet available. But well, if people really don't want to have IV therapy, then maybe it should be an option.
Yeah, and I think the discussion will come back later, maybe when we talk about registries. If such this approach that you are advocating is probably going to be used, evidence or no evidence, and studies or no studies. But it would be good to have good registries like the Go Deep registry that can follow up on those patients and really look at long-term results of such an approach. Yeah. Can I have the next slide? So then Marion Delcroix from Leuven discussed drug development with this new situation of sotatercept being in background therapy. So this makes the whole playing field for drug trials really difficult. Many new questions arise because sotatercept has a big effect. So how do you prove that that a new drug, even added on to sotatercept, is still going to be effective? Probably something like a 6-minute walk distance is very difficult to use. So different endpoints are required. Quality of life, as Louise says, is of course very important. But maybe we also need to reconsider the use of invasive hemodynamics, because in the end, this is what happens in the disease. The pulmonary vascular resistance goes up, and if we can normalize this, maybe that's something that we should really try to achieve. What is also complicated is whether in a trial, that when you design a trial, whether it should be mandatory to have sotatercept in the control arm, because this is more or less becoming a standard of care, and it could be unethical to deny a patient access to sotatercept before you start doing testing, before you start testing a new drug. But when sotatercept is only available in a couple of countries, this becomes, of course, impossible to execute a trial, and it also becomes irrelevant when a lot of patients do not have access to sotatercept. So that's a difficult discussion. So Marion went on to talk about enrichment strategies that may identify patients that so that we can do trials in a smarter way, and maybe with smaller sizes of the population. But a lot of work needs to go into that, and I think for this new work, it's very important to have the voice of patients in designing trials, and it's also very important to have a network of centers that do trials, so that we don't do useless trials. So that we really focus all our resources and all our patients to the right trials, and be effective in such a way.
Yeah, because I noticed something else during the discussions. Marion Delcroix was asked about the use of quality of life as a potential outcome measure in clinical trials. And she noted that only about 20% of her patients complete the questionnaires that they send before follow-up visits. And she suggested this might mean patients are not interested in them. Well, I don't agree, actually. I think it's quite the opposite. When patients take the time to complete these questionnaires, but their answers are not acknowledged or discussed during consultations, they begin to feel that their input isn't valued. And this can lead to frustration and lower response rates over time. So if used properly, these tools can be incredibly valuable. When healthcare professionals actively engage with patients about their responses, it shows that their quality of life matters. And that can motivate patients to stay involved. And then can be a highly relevant and powerful tool for use in clinical trials. They can provide insight into how patients are truly doing beyond clinical parameters and help evaluate whether a treatment is making a real difference in daily life.
That's an excellent point, Louise. I really think that's extremely important. But it can be quite cumbersome to fill in all these lists, and if nobody looks at these data, then subsequently, of course, you stop filling them out. So it's an extremely important point. Next slide, please. Oh, yes, this one. Yeah, this is a discussion that is ongoing in the research field, and that's the discussion on disease modification. Disease modification is a term that was used in rheumatology first, and now this term is also making its way into trials for pulmonary hypertension, and there can be some discussion on the use of it. Can I have the next slide? So Mark Toshner from Cambridge tried to explain this concept. So in rheumatology, it was quite clear. In the early days, there were only painkillers basically for patients. So as long as patients used their painkiller, they didn't have symptoms. But the destruction of the joints would just continue. The inflammation of the joints would not really be countered, and therefore the disease would just take its normal course. Well, this concept is a little bit different in when you talk about disease such as pulmonary hypertension. Of course, we have a disease process that works on two fronts. Basically, we have the changes in the pulmonary vasculature, and we have the development of right heart failure. So you could modify the disease in both areas, but it's also quite difficult to really prove that you do disease modification, because we lack the proper tools to really assess the primary pathobiological changes in the disease. We have no real imaging tools that can really tell you what's going on in the pulmonary vessels. When we do an NT-proBNP or an exercise test or imaging of the right heart, we get indirect information and not real direct information on the disease process. So it's difficult to define disease status in an individual patient. So there are some ideas about trial design. How can you show that a drug really does disease modification? And that's shown in the little diagram on the bottom of this figure. So the dotted line more or less presents the disease when there's no treatment, when there's just a natural course of the disease, and the upper line reflects the course when a treatment is given. And one of the ways to show a potential disease modification was the purple rectangle. If you stop the disease, if you stop the treatment, and you end up at a functional level that is just the same as somebody who would never have who never received the treatment, there was no disease modification. But if the disease continues to go on at a slower pace, even when the drug is being stopped, this could signify disease modification. Well, the concept is nice, but the withdrawing treatments is of course not so nice for a lot of patients. When the first treatments for pulmonary hypertension became available, some of these withdrawal trials were actually done with very poor results. Actually, it was deemed to be unsafe. The situation now is a little bit different because patients have a lot of background therapy, so maybe it's safer to do a withdrawal trial. But still, it's difficult, particularly because we lack the real good tools to assess the disease status at the level of the pulmonary circulation. Louise, I think I can get your opinion. The next slide.
Well, the patient's perspective, but that's not really about modification, but about being in remission, because it was also discussed, I think, in this presentation by Mark Toshner. And from a patient's point of view, remission is not just a medical term. It represents the possibility of reclaiming a sense of normal life. It means stability, fewer symptoms, and most importantly, way more freedom. And patients often describe remission as the ability to plan ahead again, to participate in daily life without constant limitations, and to reduce the emotional and physical burden of living with pulmonary hypertension. While survival is, of course, important, patients want to do more than just survive. They want to live just like anybody else. And therefore, achieving remission should not only be defined by improved test results, but also by improvements in energy, mobility, mental well-being, and social participation. These are outcomes that truly matter in daily life. So that's not really about modification, but in the end, well, there should be a sort of state of remission. So
yeah, and I think that this is you could also if you could approach it in the same way, use a quality of life parameter as a study endpoint, and then with a withdrawal study show whether or not this treatment has a persistent and enduring effect on quality of life even when after you stop the drug. But still, the same, I think, the same reluctance to withdrawing a drug would remain. Yeah, but it's a difficult concept, disease modification, and I'm at this point I'm not so sure whether it's going to be extremely helpful in drug development. Next slide, please. Yeah, this is just to show that a lot of there was a nice presentation by Sudhar Rajagopal from the United States, showing that there is a lot of work ongoing on determining really the primary disease process in the lungs with techniques such as PET scanning, or in this particular example on the right, it's hyperpolarized xenon, a gas that you can inhale that is being taken up by the lungs and gives you an idea about the airways, the alveoli, but also the blood vessels. So that could lead in the future to a better assessment of the effect of drugs on really the primary disease process, on the remodeling of the pulmonary vessels. And other techniques are focused on the heart. On the left lower panel, you can see an image of 4D flow MRI, a novel MRI technique that gives much more information about the function of the heart and could maybe ultimately replace a right heart cath. So work is ongoing on having good measures of the disease process, but there's still no formal validation with the really important things, as the FDA always states, an endpoint that reflects how a patient function, feels, or survives. Those new imaging techniques are not there yet. But it's appealing and promising, and I think we will hear a lot about this in the future. Louise.
Yeah, I think this is all very great, because what if it means that repeated right heart catheterization is no longer necessary? That's so nice. So go on with these techniques and use AI and everything that's possible, and I hope validation will be done in the future, because this is very hopeful for us.
Great. Yeah. Next slide, please. Yeah, next slide. Yeah. So then we had a topic that almost prevented all the American participants from coming to Amsterdam, because these terms are controversial now in the United States, diversity and inclusion. But we thought it was very important to discuss. But simply having this in an email, these words in an email, is for some American investigators now a liability, unfortunately. This topic can be addressed from many different ways, and there are many different aspects to it. But we decided to address sex differences and representation of patients into clinical trials. Can I have the next slide? The next slide, please. Yeah. So Deimante Hoppenot from Lithuania was also one of the organizers of the symposium, took on the task of discussing sex differences. For a long time, it has been known that women, of course, are more likely to develop pulmonary hypertension, particularly pulmonary arterial hypertension, but men are much more likely to die from the disease after the diagnosis. They seem to survive poorly in comparison to females, but there's no clear explanation for either of these two statistical facts. Of course, sex hormones are likely to be implicated, but drug trials that were done that really addressed sex hormones in pulmonary hypertension were not very positive. So sex hormones don't seem to be suitable treatment targets. The other aspect is, of course, the fact that different genders seem to respond different to therapeutic drugs. There are some sub-analyses of large clinical trials that have looked at this and have suggested some differences in responses to, for example, PDE5 inhibitors. But this area is still very much underexplored, and there's probably still a lot of data out there that was never analyzed or was never made public. So a lot of this still needs to be done. And it has led to the fact that gender differences are currently not accounted for in therapeutic guidelines. So a lot of improvement can be made in this field. Louise?
Yeah, that's why I added next steps, because, well, discover treatments designed for each sex would be nice, just to find out the difference in how PAH develops and responds to drug in men versus women, and also include sex as a key factor in all clinical trials and treatment guidelines, ensuring that doctors choose the best therapy based on whether you're male or female. More personalized care, actually, and hopefully fewer unwanted side effects.
Yeah, absolutely. So it's a matter of thinking about this in future clinical trials. But I also, as I said, I also think there's already a lot of data out there that was never properly analyzed, and a lot could be gained there too. Next slide, please. Yeah, and then Katie Brown Johnson from Stanford University gave us her perspective on promoting inclusion of underrepresented patients into clinical trials. So she doesn't work in the field of pulmonary hypertension, but mainly in less rare cardiovascular conditions. But she's doing very interesting work that is aimed to include more patients into trials, and particularly in the United States. It's a very clear point that, for example, African Americans or Native Americans are recruited into trials in about 60% less than other ethnic groups. And of course, this could lead to conclusions on the benefit of drugs that are not applicable to all patients. So she talked about the possible explanations for this underrepresentation. In part, it's systemic racism, and in part, it's also mistrust from minority groups towards the healthcare system. But there are also ways to improve representation. And the approaches that she has been studying was to do community engagement. That may not be the most effective way in a rare disease. It's probably much more effective in a condition such as systemic hypertension or diabetes. In a rare disease, it's more difficult. But other ways are also available, such as making better use of registries and social media. And these approaches may take away some barriers during recruitment. One of the difficulties is related to the point that I discussed previously. For example, sotatercept is a drug that is on the market in the United States, but still very expensive, and I think not available to many patients because of co-payments and caps to their reimbursement over a year. So if you now in the future are going to require being on sotatercept as a background therapy for enrollment into a clinical trial, you would automatically raise or augment the degree of underrepresentation of minorities into clinical trials. So we have to really think smartly about this and make sure that this problem is not just only becoming bigger. Next slide, please.
Well, the only thing I wanted to say is to better include underrepresented patients in cardiovascular clinical trials, we should look at a unique role patient associations can play. But I think we are way out of time, so I want to say a thing. I don't know. Yeah, I think we should stop it now, actually. So is that correct, Robert?
Okay.
No, please finish up. We have at least 5 or 10 more minutes.
Oh, okay, super. Well, I noted some dots, but I think it's something to consider in rare diseases. Yeah, in this case, I think we can help. We can really help as patient associations.
Yeah, I think that's a great point, because like a lot of her work was focusing also on community-based, having a community ambassador living in the community who could help reaching candidates. That is something in a rare disease which is extremely difficult, because how could you have an ambassador in every community for a rare disease where in many communities there will be no patients? But this particular role is really tailored for patient association. So that's a point that she didn't discuss, but I'm very glad that you raise now. Yeah. Okay, next slide. I think we will stop after this probably. Yeah, after this discussing this, I think that's we've had already a lot of good points. So I think it's also becoming too much otherwise. So the session 4 was on global aspects of drug development and availability, and the first talk was on the Go Deep registry. Can I have the next slide? So the Go Deep registry is a registry that was really set up by the PVRI. And its aims are to prioritize and facilitate global drug trials, amongst others. It's now a meta registry, as it's called. So it's a combination of different registries, with now including 32 pulmonary hypertension centers across the globe. And many centers are still looking into participation. In the trial are now almost 40,000 patients, so that's an enormous amount. There's a lot of discussion on the quality of the data when there's a lot of missingness, so to say. So some variables are not available, and all these registries are, of course, set up in a different way. It's not always clear whether patients were, for example, receiving a right heart catheterization before they were entered into the registry. So there's some discussion on whether the data are really representative, but at the same time, because now we arrive at such high numbers of patients, you can do a very good subgroup analysis, and you can account for the missingness of data, and therefore get very meaningful insights from such a meta registry. And it could inform on improving recruitment strategies into clinical trials. It could also enable the performance of registry-based clinical trials. So that's not a formal randomized clinical trial, which is, of course, extremely expensive. But you could also do a registry-based clinical trial. For example, for one of those questions that Louise raised initially, is it possible to use sotatercept in a treatment-naive patient in high risk? This is a rare situation, but we will see in the future that patients will be treated with sotatercept, and a registry like the GoDeep registry could then be used to mimic a formal clinical trial just on the basis of the data. The GoDeep registry is also collecting other novel biomarkers, blood biomarkers, imaging biomarkers, and analyses of the data could be used to improve enrichment of trials, make sure that you do the trial in the right patients, and also make sure that efficacy is assessed in a proper way. And something that could be useful for pharmaceutical companies, it could also aid in calculation the costs of a particular trial. Louise, next slide, please.
Yes, I already see it. This registry can really help improve how clinical trials are designed. For example, it could help researchers decide who should or should not take part in a trial based on real-world data, and make sure trials include enough people to show whether a treatment works, and test if new digital tools like wearables or apps can track disease activity. Because the data follows patients over time, it may also help predict how the disease will progress and which treatments give the best results for both health and costs. So that's a nice combination, I would say.
Great. Next, then we had a pro-con debate on whether big pharma should focus on making the current drugs available for all patients, or should focus on drug discovery. Well, actually, it didn't become a very heated debate. I think we all more or less agreed that drug availability is a big problem, but it's not just a problem of the pharmaceutical industry. It's a problem of all of us, basically. On the left shown the size of the different populations in the world, and then the big balloons of China and India, where a lot of the drugs that we've been discussing over the past 30 minutes are not available to many patients. So there are a lot of things that are necessary to reduce barriers for drug availability. But at the same time, the primary responsibility of big pharma is to really develop new drugs. They're not philanthropic institutes. Of course, it's the way that the system has been set up. Our governments have decided that the drug development is very expensive, so it's better to outsource that to commercial parties. And as such, we cannot expect that they will truly focus on drug availability only. But at the same time, we can, of course, urge them to be at least reasonable in how they set their prices, and also how they can think about not doing really harmful things like just evergreening patents to make a drug seem new, but in fact the drug is still the same as the old drug. These kinds of approaches are really not very helpful. Can I have the next slide?
Yes, well, actually, I agree with what's said. The discussion was actually the conclusion from both medical experts was clear. Innovation and access are not opposing goals, but they are twins. And actually, as a patient representative, I couldn't agree more. So that's actually what's said underneath.
The fifth session during the conference was about innovations in trial design. The session was moderated by Louise Bouman and Francis Varian. And Louise Bouman kicked off with her first presentation on how risk scores can be used in clinical trials. Louise has done a lot of research in the past years, developing and validating different risk scores to assess patient risk. And mostly these risk scores are currently being used in the treatment algorithm. But these risk scores can also be very useful in designing trials, either to enrich patient populations, to really zoom in on specific patient groups and to see whether adding treatment or escalating treatment is useful. And risk scores can also be used as trial endpoints because usually they correlate quite well with the relevant endpoints for patients. Risk scores tend to correlate very well with survival, obviously. Therefore, there's a lot of discussion ongoing with FDA and EMA to see whether risk scores can be acceptable as trial endpoints. Another very attractive endpoint for a clinical trial could perhaps be found in imaging. In recent years, we have seen a lot of development in imaging of the right heart. We all know that right heart function is very important in patient outcome, and different parameters correlate with survival. And therefore, a lot of people advocate for the use of using right heart imaging endpoints into clinical trials. Still some validation perhaps needed, but it already, at least for a proof of concept study, RV imaging seems very useful. Pulmonary imaging in the field of pulmonary arterial hypertension is still quite in its infancy. There are several machine learning or artificial AI approaches to quantify the degree of pulmonary vascular disease in PAH. These efforts look very promising, but still a lot of validation is still needed. Most of these risk score endpoints and imaging endpoints are being considered in larger randomized controlled trials, but we can also think of using them in much smaller trials. For example, in really small trials, N of 1 trials, or withdrawal studies, and Martin Wilkins discussed this topic. He showed very interesting data using a CardioMEMS device, so an interpulmonary pressure monitoring device, to look really fast, real time in changes in hemodynamics in patients treated with PAH specific drugs. And because these devices give so much detail, continuous measurements, they can show you on-off phenomenon when patients are being treated with PAH specific drugs. And this could lead to, with very smart trial designs, to much smaller studies and much better identification of proper dose levels with good therapeutic efficacy and without side effects. And so this talk by Martin Wilkins was also very interesting. Other statistical approaches can also be contemplated to reduce sizes of study populations. These can be quite complicated use of Bayesian statistics, but with the big advantage that trials can be continuously monitored, and at an earlier time point, it can be determined whether a trial has recruited sufficient numbers of patients, and therefore trial sizes can be reduced using Bayesian statistics. And the other topic of the win ratio is also interesting. In the past years, we have seen several trials in which a composite endpoint was used, such as time to clinical worsening. For example, a Griffon trial, such an endpoint was used, but there has been some critique because in this composite endpoint, it is sometimes occurs that, for example, death of a patient or hospitalization are almost treated in a similar way. Whereas, of course, the meaning for these events is very different for a patient. And the use of a win ratio can change this a little bit by prioritizing certain events over others, and therefore coming to a more meaningful clinical endpoint. And then I gave a talk on practical and ethical aspects on open label extension studies. Open label extension studies have more or less become an industry standard. After phase 3 randomized clinical trials, usually patients can be enrolled in an open label extension study. So all the patients, even if they were on placebo, get active drug. So in terms of drug availability, it's very attractive. It also provides a means to continue drug treatment in patients that were on active drug and benefited from the treatment. And open label extension studies allow a much longer monitoring of patients, and therefore much more safety data can be collected. So all these things look very good for an open label extension study, but there are also some disadvantages. One of the disadvantages could be that patients are no longer available to participate in another study as long as they are part of the open label extension study. You can also question the validity of the safety data, because most of these studies look in a subgroup of patients, and patients that actually had no benefit or had a lot of side effects from active study drugs will tend not to participate in the open label extension study, and therefore the results of such studies may look much better than the actual results really are. Open label extension studies are very expensive, and it could be maybe preferable in some instances to make sure just that all the study participants have access to the study drug after the end of the study without performing a formal open label extension study. So patients in such a scenario do have access to a drug without all the complexity of being in a real formal study. And safety data can be collected in a real world registry. So there can be some discussion around this, and the conclusion, I think, from this talk was that even though open label extension studies are very attractive, we should not just consider it as a default, and we should always think really well whether this is something we really want to do or not. And then the final talk was on the climate impact of drug development, given by the other chair of the session, Francis Varian, with very thought-provoking and impressive data on the impact of clinical trials, not just making a placebo, but particularly also the travel of patients to clinical trial sites has an enormous climate impact. And Francis talked us through this and also gave a lot of alternative options within clinical trials, maybe reduce the number of patient visits or find a way of remote monitoring of patients, which can have a dramatic impact and really improve the sustainability of a clinical trial. Louise, you can continue.
So I wanted to go back to the talk of David Keeley about the use of, for example, CMRI, cardiac MRI. And there has been a recent meta-analysis of 20 studies involving over 2,000 patients, and that confirmed that this kind of MRI can reliably predict outcomes like mortality and clinical worsening in PAH. And this is really very encouraging from a patient's point of view, because if a non-invasive method like an MRI can provide such strong prognostic information, I really am sure that most patients would absolutely prefer it over an invasive procedure like a right heart cath. CMRI is safer, more comfortable, and less stressful. And yet it still gives valuable insights into how our heart is coping with PAH. So I think these kinds of things are terrific, actually. We think as a patient group that this should be more widely used, not only in research, but also in clinical practice. to help guide treatment and follow-up in a patient-friendly way. And the innovations like AI-enhanced imaging and multimodal techniques like implantable devices can offer detailed patient profiling and quicker, cost-effective measurements. So I have really a lot of hope for the future for comfortable, how do you say it, guidance actually. Next slide, please. Yes, thanks. This is the discussion you talked about, Harmjan, the open label extension. And I think for the patients, the option to continue treatment after a clinical trial, so the first step in a clinical trial through an open label extension study, is a key reason to participate in the first place. Knowing that they can stay on a medication that proves effectiveness for them and others, provide reassurance and motivation to join. If this option were no longer available, it would likely discourage participation and not increase it, actually. Those open label extension studies provide long-term data on safety and effectiveness, which is also needed for national and international authorities, and other ethical benefit like ongoing treatment. Alternative approaches like what you called early access programs or delayed start trial designs might offer similar benefits, but they must still guarantee access to treatment for responders. For patients, continuation of effective therapy is not a bonus, it's a priority actually. And the next slide, please. This is about the talk of Francis Varian about climate impact of drug development. And what I liked about it is indeed the traveling of patients has an enormous impact on the climate. So if we change that with using digital tools and decentralized trial designs, we can reduce the environmental impact of clinical research. And this approach can lower emissions by 20 to 30%, while also making it easier for patients to stay in the trial. So it's a win for the planet, but it's also a win for the patient. So I'm very positive on this thing too, actually.
Yeah.
Next slide. Well, then this was, I think, oh, this is you again, I think, yeah.
Yes, I think so too. The final session of the symposium was a summary of several developments in the treatment of pulmonary hypertension, not just new drugs, new molecules, but also new devices for the treatment of pulmonary arterial hypertension. And the first talk was by Professor Jing from Guangzhou in China. He has been working on an anti-based therapy for pulmonary arterial hypertension, but actually the talk that he showed us was a little bit different. He introduced us really to the great work that has been done in China. A big PH center in Guangzhou and also large collaborations within China to study pulmonary arterial hypertension. A lot of new findings are expected to come out of China, not just because new drugs are being developed, but also because there are very large cohorts of patients and a lot of biological samples are being collected. And Professor Jing invited all the other scientists in the room to collaborate with Chinese partners and to use, for example, all the resources in China as for example validation cohorts for finding new biomarkers. So that was a very interesting start of the day. The next talk was done online by Roam Zemanian from Stanford University, who talked about the development of Alephine for the treatment of pulmonary arterial hypertension, mostly building on decades of work actually by Marlene Rabinovich in Stanford, showing that elastase, an endogenous enzyme, is elevated in pulmonary arterial hypertension and contributes to progressive remodeling of the blood vessels. And allophrin is a natural inhibitor of this elastase, and can be made and developed into a treatment option. And the Stanford group has already done quite some preparatory work for a randomized clinical trial, and is now quite advanced in getting funding from the NIH in the United States to be able to do a full-scale clinical trial. So we hope that despite all the difficulties with funding in the NIH funding in the US, that this study will be possible to be executed. The next talk was about mosliciguat by Professor Gofrani from Gießen. Mosliciguat is a guanylyl cyclase activator, so a little bit similar to riociguat, inhibitor of riociguat. Mosliciguat is very powerful, a very powerful vasodilator with very promising phase 2 data, and now the drug is being developed for inhalation in patients with pulmonary hypertension in interstitial lung disease. So we expect a lot of news in the next year or so about the efficacy of this drug. Then the next talk was by Paul Yu, who really from Harvard University, who was really very at the start and of the development of a whole new class of drugs, activin inhibitors. Sotatercept, of course, a very promising new drug, very positive results in phase 3 studies. But Paul Yu told us how the development of this drug came to be, and also how complicated it still is to understand the action of the drug. But also that understanding this action is really important, because it could also lead to new drugs in this class, which could also be very effective and perhaps have fewer side effects. After the break, Professor Pilar Escribano from Spain, from Madrid, talked us through development of seralutinib. We've already seen a very interesting phase 2 trial with this PGF receptor inhibitor, and now data are collected in a phase 3 trial, and we hope to hear soon what the efficacy is in the phase 3 trial of seralutinib. Then we went on to devices. Mark Pritzer from Minnesota talked us through the development of the so-called ARIA device, a complicated device that adds compliance to the pulmonary vasculature. So in a normal heartbeat, when the heart is ejected out of the heart, a lot of the energy is stored in the vessel wall of the pulmonary arteries. And at the end of every heart cycle, a lot of this energy comes back into the circulation and propels the blood further into the lungs. This compliance is much reduced in patients with pulmonary arterial hypertension. The vessels become stiffer, and Pritzker and his colleagues have developed a device that mimic the normal compliance of the pulmonary vasculature. And even though this is still in development, it seems to really help right heart function and could prove to be useful in the future. Finally, there was a talk by Alex Rothman on the use of pulmonary artery denervation. That's a catheter-based approach to cauterize them, or to use electrical current to destroy small nerves that are located inside of the vessel wall, or in the wall of the larger pulmonary vessels, and contribute to a high pulmonary vascular resistance. This device can be, or this approach of denervation can be used to lower pulmonary vascular resistance and is now currently explored in other forms of pulmonary hypertension, not in pulmonary arterial hypertension. But if the results are promising, we hope that this can also be applied in pulmonary arterial hypertension. Louisa, I will.
Yes. Can you give me the next slide?
Yes, I'm trying.
Oh, doesn't work. Well, it doesn't matter, but
it's there. It's there.
Yeah. Oh, yeah. Well, seralutinib and mosliciguat are both inhaled drug. And I remember we talked about it together, Harmjan, afterwards, after these presentations, because there was a discussion about if it would help and if the medication in both ways were going to reach the area where it should work, and also about the side effects, because it gives some sort of, at least for seralutinib, cough. And then I said to you that I'd rather have a cough than being nausea or having diarrhea and other stuff when that happens when you give such a medication in a normal way, so an oral medication. So that's what I wanted to say, that for patients, I think it's way easier to do inhalation without the mega impact on your bowels, actually. And hopefully it does work, because I think the seralutinib shows enough benefit at least to do a phase 3 trial, and also a new one for PH-ILD. So that's just something I wanted to comment. And also for mosliciguat, it's only an inhaled drug once a day. So that's also, wow, progress, I would say, because then you don't forget it so easily. So.
Okay, that concludes the discussion of the meeting. I think yeah, we can come up with 3 important take-home messages. First of all, the availability of sotatercept will really change many aspects of not only of the treatment of current patients, but it will also change drug development. We'll see new kinds of drugs being developed that could have an add-on effect on sotatercept. We will have to develop new treatment algorithms, and we'll have to define the place of sotatercept. Maybe in a patient with sotatercept, we need to think about new trial designs, new, more efficient trial designs, different endpoints using different biomarkers. There's still a lot that we need to figure out, and how sotatercept will be available for our patients. Our meeting was also, in a way, a call to action to address gender differences and barriers to trial participation, to global disparities in drug availability. And yeah, particularly also, I think today's online presentation really shows you how important it is to have patient participation in drug development. So many important things that are being brought up by patients, and I really appreciate Louise's participation in this talk, because I think it shows that drug development is really a collaboration between patients, healthcare providers, and pharmaceutical companies. And only if we do this, the 3 of us together, we can really make a useful contribution. Yeah, just something personal. I think a small meeting such as this drug development symposium are really nice. You get to talk to a lot of people. You're not just lost in a big venue with a lot of different parallel sessions, but you really are focused with the same group of people, and that was really nice and was really valuable. And the weather was nice. I think a lot of people appreciated being in Amsterdam, which made me feel a little bit proud, because I love that city too. Thank you all very much, and I'll hand over to Louisa for some final words.
Thanks. Yeah, I can only agree with what you said. It was a very nice meeting of 2 days, and indeed you could talk to people during a longer period. So I got to know a lot of people. And so as you can see, I don't think this was the whole group, because a lot of people were still taking a water to drink, because this was just afterwards. So everybody was thirsty because it was very hot. But a big part of the group was on the picture. So we had a lot of fun, actually, because during the sessions you're pure serious, but afterwards you can have a little bit of fun with it, which is also important. And I totally agree with what you said about a new development, that I think it's very important that patients are allowed to think together with the pharmaceutical industry and scientists and caregivers about why drug development is important, but also which kind of drug and with what kind of purpose it's developed. So I just can say it was very important to be here, and it's very nice to hear all these new things, although some parts were pretty difficult, and it's also too difficult to explain. But it's very nice to see that there's so much development, and it will give us a lot of hope and courage, I think, for the future. So thank you all for listening, and I hope it was encouraging to you too.